Good morning and welcome to our talk today. My name is Jason Amsbaugh, and I'll be your moderator. The title of our talk is Accelerating Cell Line Development from DNA to MCB. Our speakers today, Louise Lindbeck, is a principal scientist, cell line development at AGC Biologics Copenhagen facility, where she leads development lab teams and customer projects, as well as research and development related activities for upstream development. Her experience and research have been focused on cell line engineering using CRISPR-based technologies in protein production and mammalian manufacturing platforms and timeline optimization to help clients achieve effective timelines and product yields. She has over 10 years of research experience and lab experience and holds a PhD from the University of Copenhagen. Our second speaker is Lily Lee. She's a senior scientist, cell line and upstream process development, and a multidisciplinary bioengineer with a decade of research and lab experience. She leads mammalian cell line development and upstream process development projects for clients at the AGC Biologics Seattle site. Her expertise includes vector construction, transient and stable DNA transfections, initial and final clone assessment, as well as process optimization using DOE bench-scale bioreactor experiments to ensure process robustness and scalability in manufacturing. She received a PhD from the Voiland School of Chemical Engineering and Bioengineering at Washington State University. All right. Well, with that, we'll go ahead and begin our presentation. And I'm going to hand it off to Louise Lindbeck to get us started.
Thank you for that. So, thank you. So I think we will just dive straight into it. So I want to just start off by just touching on why we're even here. So there's currently a growing demand for cell line development activities, and the market for CLD services is expected to reach $1.7 billion within the next 5 years. So in this growing demand is a request for bispecifics and monoclonal antibodies, but also recombinant proteins and vaccines as well. And despite this increasing demand, CLD remains technically and operationally a quite difficult task. All the while, there is an increasing demand for shortened timelines. So this is where CDMOs come in. We're contract development manufacturing organizations like AGC, where we collect latest techniques and technical expertise, as well as the lab space to tackle these complex questions and demands from customers, all the while shortening our timelines. So I'm going to go straight into the first case study. So Lily and I will be presenting a number of cases, and the first I want to talk to you guys about is the attempts and the work that we've done to fasten the CLD timeline at AGC. So at AGC, we do our whole transfection work from start to end, including MCB in-house. And there is a major benefit to this process, especially when it comes to timeline optimization, because it leaves us in charge of our own timelines. So obviously this would be a benefit when trying to shorten them, but also just meeting timelines in case something has gone differently than planned. It is easier to catch up on lost time. So taking you through some of the process of, I'm just going to get this away here. So taking you through some of the CLD process that we do at AGC, it would involve first designing and making the DNA, of course, but then when we're ready for transfection, we would thaw our in-house DG44 cell line. This is a CHO cell line that we use for production. We would transfect it with the DNA of interest, and we would create a stable transfection pool for two main purposes. First, we would inoculate production models in shake flasks. This is a process that would run for approximately 14 days and allows us to evaluate titer and product quality. But we'll also use these stable transfection pools for single cell cloning. Now, single cell cloning is important, obviously, for generating monoclonal cell lines. And we disperse the cells, the stable transfection pool, into 96-well plates, which are then imaged over the course of 14 days. And these images are used for clone selection. We would perform a monoclonality review, looking at all of these many pictures to select 400 clones for expansion. Once we have expanded them, we can sample for titer, and this titer information is used for clone selection and expansion into shape flasks. So obviously we don't expand all 400 into shape flasks. So these titer information, we would select top 30 that we then move forward into production models, allowing us again to get a picture of the titer capacity of each of the 30 clones, but also product quality that we then use for top 3 clone selection, moving forward into a research cell bank, but also evaluation in bioreactors. And now this brings me to a quite sort of important milestone in cell line development, which is the RCB. So the RCB, the research cell bank, is this largest cell bank used to move forward, and that serves the basis of the MCB. And the reason it's such a milestone, especially when looking at timelines, is because there's a sort of a list of quite time-consuming workflows that follows the RCB generation. So firstly, not super time-consuming, we would sequence the gene of insert. Now this is done, the work is done in-house, and the fragments are then sent up for sequencing. But the stability study is super time-consuming. So the stability study is this prolonged seed train where we use our RCBs, we passage them over the course of 9 weeks, and then we evaluate their titer capacity. Also, we'll send our RCB for release testing in order to thaw it for MCP production in our GMP facilities. Now, these two last points are rather time-consuming and not very easy to change, kind of out of our control, basically. So in an AGC standard process, we basically go from transfection through pool generation over RCB generation to MCP. And the way we do this in a standard protocol is that we continuously narrow down the number of clones we look at. So we take one step after the other, narrowing it down before we move on to the next step. Ultimately, we would bank top 3 clones in an RCB, and that would take approximately 12 weeks. Now, what we also see here in a standard timeline is the rather long stability study. So again, this is not very easily changed, and that means that activities associated with shortening this timeline would typically be the activities that go before the RCB generation. And one example of these steps that we look to evaluate and that we look to shorten is the clone expansion step. So looking at this clone expansion step in a standard process in the industry, this would include some quite time-consuming and also laborious work flows. Typically, what people will do, and what we used to do as well, is to take our clones from the 96 well plates and expand them through a number of different well formats. Typically, we would go into 24 well plates through 12 well plates before moving the clones to 6 well plates. Now, this would take a long time, and it would require quite a lot of handling as well. So what we did when setting out to accelerating our timelines was to just do a very simple setup. Basically, we seeded a fixed amount of cells into these different plate formats, and then we waited for 7 days, and then we counted them, kind of to simulate, I guess, a cell expansion standard workflow, where we would remove the clone. But what we found was that moving the clones from the 96 well plates straight into the 6 wells generated a significantly higher number of cells within 7 days than we would have if we'd gone through these other plate formats. So this is an example of how we performed this flow optimization, basically saving ourselves 2 weeks, but also a lot of handling. And overall, we could achieve a decrease in 15% in the overall CLD timeline, all the while saving handling, but also sort of decreasing risk, because of course with every flow of cell handling there is a risk of contamination, especially when working in plates. So this was a major benefit for our optimization process. Now, if we go back to the overall cell sort of CLD process, in a perfect world, I think one might imagine that going from transfection and single cell cloning to MCB, if we could do that in the time it would take for one cell to become sufficient cells to bank in a master cell bank, obviously that would be wonderful. But of course, it's not really doable with the expansion and the shake flask production models of 400 clones. So any optimization step would be a sort of compromise between the amount of work that we're able and capable of handling in the lab and the cell proliferation and wait times. So apart from looking at the expansion steps, another focus point in developing an accelerated timeline was the wait times. And in a sort of attempt to decrease these wait times, we had a look at when we generate the RCB. So again, recall how the RCB generation is really a milestone, because the time-consuming step of the stability study comes after. So the earlier we can move this up and do the other work simultaneously, the better. And what we proceeded to do in our standard workflow, apart from shortening our expansion step by moving straight to 6 wells, was to not wait for the decisions to be made and rather just bank our top clones in an RCB as early as possible, getting a head start on the time-consuming downstream activities in terms of RCB testing, but also the stability study. And then in the meantime, doing these quality analysis. So basically, overall, these velocity activities allowed us to shorten our RCB timelines from 12 to 8 weeks. And we can now provide MCBs to our customers within 20 weeks. Overall, this means that we have a platform that we can offer customers that provide them with a drug substance within 12 months. So all the while, time being a major issue and a major factor for our customers, another very important part of being in CDMO is the ability to be flexible in the workflows we do and to help provide the expertise to solve sort of complex issues. And one example of this is the case study 2 I want to take you through on post-cell engineering. So in this, background is that we had a customer at AGC that had come to us for cell line. We had developed a clone for them and that they were technically happy with. But what we found was that during the production process in our CHO cell line, the customer's protein would co-elute with the CHO host cell proteins. So basically, this is not abnormal. CHO host cells would produce lots of CHO proteins, and these do sometimes bind the customer protein. But it is the job of downstream then to find a way of separating these, because of course we can't inject these drugs into people holding a lot of CHO protein as well. The issue in this specific customer project was that the downstream process development was stalled because of this co-elution. So basically, we couldn't separate them. So what we did was that we employed gene editing techniques to delete the host cell protein that was believed to be the main problem. And within just 3 months, we had a new RCB created for them. So I'll just take you through some of this, how we did this. Basically, we designed a number of sequences, 3 different ones, targeting the exons of the host cell protein we needed to delete. We were not looking to do a complete knockout. We just looked at the out of frame insertion and deletion percentage, because basically we just wanted to create a deleterious mutation, losing the expression of the protein. And what we found when we initially transfected with these different sequences was a quite good efficiency. So we tried different conditions. We did single transfections, we did double transfections, Some of the sequences worked better than others, but altogether we achieved, for some cases, more than 60% out of frame insertions and deletions, and we were quite happy with this. So we moved forward with these pools, and we single cell sorted them into 96 well plates. Now, this is what we do in a standard project as well, right? We would single cell sort them, we would look for monoclonality, and we would move 400 clones forward into 6 well plates. And that's what we did here as well. And in this specific setup, we then looked to identify 96 top clones for further expansion into shake flasks. But what we found, interestingly, was that the majority of these 96 top producers, they were clones from double transfected pools. So this would suggest that there is a correlation between the host cell protein integrity and the titer capability of the cells. Now, this is not technically surprising. There is literature on this. Host cell proteins take up some of the cell's capacity, but it was, of course, a very welcomed byproduct of our efforts. We moved forward with our top 30 screenings. So from these clones, we identified, well, we did a genome analysis, we found the actual knockouts. We moved 30 of the highest producers into shake flasks, and we did production models to do quality and titer assessments. And what we found was that the titers in our genome engineered cells with the HCP deleted here in green, we found a 3 to 4-fold increase in titer when comparing to the original clone selection efforts that were done with the mother clone. So again, confirming that deleting these host cell proteins was, or this specific host cell protein was associated with an increased capacity for titer. Ultimately, the same pattern was seen at the end of the stability study. So stability study, just to outline here in the green figure, involves the thaw of an RCB, passaging for 9 weeks, and banking along the way at strategic points to mimic cell banks for late-stage projects. At the end of the stability study, these banks are then thawed, they're run in production models, and titers can be compared. And what we found was that the mock cell banks here, and the titers here in the top, in the purple part of the graph, of these newly generated top clones would perform significantly better than the top clones from the original run here below. So in conclusion, we demonstrated how important it is for CDMOs to display this flexibility in our workflows that allows us to customize our CLD campaigns so that we can solve the customer's problems, whether they're from outside of our company or from projects we're generating within. And in this specific case, with the added benefit of increased productivity. So with that, I will hand the word over to Lily.
Hi, everyone. In order to further elaborate on the flexibility and customer-driven nature of the COD workflow that Louise has talked about, here's a case study that also demonstrates the ShareOne platform success with different molecules types, including those that are difficult to express. In this example, an early stage client, customer Y, initially came to us with sequences of 6 different dry-specific protein variants and requested that we quickly generate some research-grade materials for variant screening and internal testing, both at AGC Biologics and at the client site. Initially, the lead variant was not determined, so it was important that all the variant materials were generated in a timely manner so that the variant screening activity could be initiated right away. It was an uncommon and also a challenging request due to the fact that the preliminary transfection titers of these variants in CHO were very low. This suggested that these dry-specific variants were difficult to express. As we all know, production stability is required for process robustness and scale-up success. However, in this case, the development of a production cell line for the lead variant would be quite challenging because the production instability would be quite likely due to the difficult to express nature of these molecules. Anyway, using the SHEP1 expression platform, we not only did we quickly provide the client with sufficient variant materials for variant screening work early on, but we also successfully generated a stable and high producing cell line for the lead variant and a production process with excellent scalability. In the next few slides, I will go into more details on each of these project outcomes. In the beginning, each of the 6 variant sequences was incorporated into the SHEP1 expression plasmid. A total of 6 different transfection pools corresponding to 6 variants were generated in duplicates. The cells were then let to recover and undergo metabolic selection using our proprietary selection medium. The viability graph that is shown here demonstrated the transfection and selection recovery process of the 6 variants. Starting from week 3, the transfection pools were expanded for a 14-day fed batch production using the SHEP1 production platform. The cultures were harvested at the end of the production, and the clarified harvest fluid for each of the variant was purified using one-step purification. So overall, from DNA transfection to purification of the pool materials, it took a total of 5 weeks. As demonstrated by this titer graph, again, all the pool titers were higher than the average preliminary transfection titer, which is shown as the red dashed line here. Therefore, and once again, we successfully generated good amounts of the variant materials for internal testing in a timely manner, which met the customer's expectation. At the end of the variant screening work, a lead variant was determined using the SHEP1 expression platform and various clone screening steps. The lead clone of the lead variant was chosen based on productivity, growth, bioreactor performance, as well as product quality attributes. As illustrated by the titer graph here, the final clone titer increased by 8-fold compared to the pool titer, which was quite significant. This 8-fold titer increase was even further enhanced in bioreactors later on during upstream process development. Needless to say, the customer was very pleased to see this outcome. The lead clone also demonstrated great production stability for at least 70 generations starting from the research cell bank or RCB level, which is demonstrated in this schematic and the titer graph. The schematic shows how different mock cell banks were generated following RCB thaw over a period of 9 weeks of the cell culture passage. Then when the RCBs and all the mock cell banks were thawed, expanded, and evaluated in a fed batch production, we saw that the production titer only slightly decreased by less than 5% from RCB to mock EOP levels. The lead clone also exhibits excellent scalability from AMBER 250 or 250 ml, 15 liter, 100 liter in process development to 500 liter in manufacturing. Here are the overlay graphs of some of the process trends across different bioreactor scales. The legends of these graphs can be found at the bottom. Cell growth, cell culture health, product accumulation, as well as a few metabolite trends and chemistries are shown here. We can see that the process trends are very similar across different bioreactor scales. Now, I would like to show you a different case study where we adapted a production cell line to different chemically defined media. In this example, one of our clients, customer Z, has a commercial product that is currently produced by a process that contains a plant-based raw material. The raw material has significant lot-to-lot variability, which is a major contributing factor to process inconsistency. Due to this reason, periodic screening of the raw material lot is required, but it adds a lot of uncertainty, cost, and time to the process itself and any related planning activities. For this reason, the customer would like AGC to remove this plant-based component from the current process. Also, like any other production process improvement, titer enhancement would certainly be a bonus. For this particular project, we proposed several different chemically defined medium options and adapted the cell line to those media. We then evaluated the cell performance in a fed batch production to choose the best suited CD medium. At the end of the chemically defined media screening, not only did we identify the best medium, we also demonstrated that the production titer could be improved by at least threefold compared to the current commercial process. The initial media screening step was performed in shake flask. The cells were adapted to different chemically defined media, and the adaptation process took a few weeks, as illustrated by this viability graph. The fed batch productivity screening was performed right after the cells have fully adapted to the new media. The current process at scale is a 12-day production process due to the EOP harvest viability being a CQA. But in this fed batch experiment, since the cell viability in one of the CD media, specifically CD media number 3, was still in the high 90s on day 12. Therefore, the process could go on certainly for more than 12 days. In fact, what we actually did was we let the production with that CD media went on for 15 days in this experiment, which yielded even more product in the end. The conclusion of the productivity screen in shake class was that the best CD medium yielded a threefold increase in production titer. And of course, this production titer increase can be enhanced by further process development work. So we've been talking about all these case studies. I think it is important to emphasize that most of these case studies would not have been accomplished without the Chef One technology. There are three key components of the Chef One platform: the Chef One expression plasmid, the Joe DG44 host cell line that was obtained directly from Jason lab and has since been adapted to serum-free and suspension culture conditions. And last but not least, the proprietary media and specialized feeds. It is important to know that the Chef One platform is license-free, and it is also free of any royalty charges for its use, which is a huge advantage if a client intends to license a product after positive phase 1 and or phase 2 clinical trial results. A few other highlights of our cell line development capabilities include the extensive and diverse experience that our scientists have with different cell lines, cell types, and expression systems, in addition to the Chef One platform. Not only that we can develop cell lines, we can also generate and store cGMP cell banks in-house. So from a DNA transfection to MCB or WCB generation and storage, we can do it all. Once again, our platform is highly flexible and can be customized so that we can work with anything that a client brings us. As the Chef One platform has been used for so many products, as well as 5 commercial products, we can certainly provide full documentation for R&D filing, such as proof of monoclonality, full traceability of the raw materials, or cell line history and testing, and so on. The Chef One platform has proven to be successful with different types of molecules, from MAPs, complex glycoproteins, to bio and dry-specific molecules. With 5 products already in the market, there are at least 55 different molecules produced by the Chef One platform today. Here's a summary of the current technologies utilized in our Chef One platform. We have the Wolf cell sorter, that enables fluorescent activated single and or bulk cell sorting following transfection. We also have the cell metric, which is a high resolution cell imager. Both the cell metric and the icon analyzer together allow us to rapidly identify and screen monoclones based on titer as early as when the clones were still growing in 96 well plates. I also want to point out that cell lines that are not expressing monoclonal antibodies, for those cell lines, the octet is used in place of the icon analyzer, and the accelerated workflow that Louise has presented in the first case study will still be achievable. This is a linear representation of the SHEP1 gene cassette. The SHEP1 promoter enables constitutive high expression levels of the product without the need for any methotrexate amplification. In case I haven't mentioned this, our COD platform is fully chemically defined with many opportunities to further enhance protein titers using the high throughput AMBER system within the upstream process development group. Now, when working with different customers, we also leverage the AGC Biologics global network whenever possible to achieve our client goals and needs. We have a total of 7 facilities around the globe with specialized capabilities. For instance, for mammalian services, we have the Seattle site where my team is located, the Copenhagen site where Louise's team is based, as well as the Boulder site and the Chiba site. AGC Biologics is actually part of the AGC Inc., which was founded in 1907 with headquarters currently in Tokyo, Japan. This provides us with great financial stability, unlimited future growth, and opportunities to further expand our global services and capabilities. With that, I would like to end our presentation here, and we will be happy to take any questions.
Thank you, Lily. All right. The first question for Louise. By making shorter the screening of the clones, does it mean that all top 8 clones go to stability studies?
Thank you. Yes, it does. So we've been sort of looking quite back and forth at what would make the most sense. And actually, we would be able to. The plan is, or what we do is that we're going to be expanding all 30 for RCB, and then we're going to be decreasing that number and banking only 8 once that decision is made. Immediately then following and starting the stability study. One option would be to sort of sort through those cells. So as we get the quality information from the top 30 clones, we could select, or from the 8 clones that we evaluate in quality analysis, then we could deselect some and only move those 4 forward in the stability study, which would be less effort and cheaper for the customer. But again, when looking at these timelines and trying to shorten them, it's not really about the money. And I think it's a major benefit bringing all 8 clones through a stability study, because in the case that some are not stable, it would increase the amount of clones we would have to choose from, or we would have to move forward with at the end. So it's a major benefit to basically bring as many as possible through a stability study. So yes, we would do that.
Thank you, Louise. This is a question, I guess, for maybe the both of you working across different sites. If I bring a product or a program to AGC, will I only have access to support from a single site that we need to select?
I can answer that question. The answer is yes. You can leverage the global network that we have across the globe. In the past, we have actually had process development that were collaborated between the Copenhagen site and the Seattle site. So there were like cell line development projects that were initiated in one site and then transferred to the other site for further process development and vice versa. So that's totally possible.
Terrific, thank you. The next question, so the next question is, to what percentage did the viability drop at the moment of harvest across the different scales? So Louise, I think this was during one of your case studies, essentially where you showed the the different scales essentially in the cells from sort of small to large bioreactors?
I think this might be for
Yeah.
me, right?
Oh, sorry. Go ahead.
Yeah, let me go back and look. I think, let's see here. I believe that it dropped down to not less than 70%. I can't recall the exact number, but yeah, I'm pretty so sure that the end of production harvest was not less than 70%.
Excellent. Thank you. The next question is asking actually if you guys have examples of cell line development in microbial host cells, so non-mammalian systems.
That would be an expertise that the Heidelberg team is specialized in, microbial cell line development. In Copenhagen and Seattle, we work with primarily mammalian cell lines.
Yeah, so I think I would encourage you guys. I mean, if interested in that, reach out to the business development department and they'll help you with that. We do have the production facility, but not the cell line development facilities in Seattle and Copenhagen.
Gotcha. This is a question I think related to one of your case studies, Lily. The 8-fold increase, essentially, what does that mean in terms of grams or in terms of just what that total yield was?
I am afraid I cannot answer that. It's client data, so I cannot really reveal much.
A lot, I would guess. All right, that sounds good. All right, are there any other questions here that you guys would like to, anybody else?
So there is a question on why using CHO cell lines if MTX is not used. So I think a lot of, so generally we like to not use MTX because it's toxic, and especially in larger quantities in production, it's more complicated. So it's nice to have a system that doesn't use it. And the benefit of using CHO is that it is very well characterized in production and in cell line development. We know what to expect. It's also very robust and very stable. And it is the most used cell line in manufacturing. So there are definitely a lot of benefits to using it. Yeah.
Excellent, thank you. Here's another question. What advice would you give to developers to help select a cell line development partner?
Sorry. Okay. I can pick up on that one. So I think I would advise that you are very specific in the questions you have and make sure that the CDMO knows what you're looking for. So definitely have it, make sure that you're clear on whether it's time or money or whatever factor is the most important, and make sure you're clear about that. And then BD will help you. And you can determine which one you feel is, I guess, the right partner for you guys.
Yeah, Louise, that's pretty much what I wanted to say too. I also want to add that based on my interactions with the customers who have been interested in AGC Biologics, and including those who are who have actually chosen us to be the partner, what they were looking for from the beginning was a CRO, a CDMO that has an excellent COD track record. That means, how many successful COD campaigns that CRO has, and how many commercial products that has been in the market using that system that is implemented by that CRO. And whether that CMO has full capabilities, meaning that not just cell line development, but also whether that CMO is able to perform process development, process characterization, validation, and also manufacturing. So it'll be better to have a CDMO that can do everything from early stage support R&D filing to BLA filing and commercialization. And of course, the speed is what Louise talked about too. Most clients nowadays want shorter timelines, so they would come to us with questions about how fast we can go to bring their product to the market or to help them file their IND.
Thank you.
Mm-hmm.
Here's a, so I guess just first a quick short question. What method of gene integration is your system using?
That would be random integration.
Okay, thank you. And then a longer question. So the clones that go through the stability studies and have good quality profile, are all of those kept as RCBs? For example, if clone 1 is used for manufacturing a product used in early phase trials, can in a future clone, like in a future clone 2, essentially be used for manufacturing in the future? So yeah. I guess the question is, do they need to go through any sort of clone comparison studies before?
Yes. So no, well, any clone can technically be used, but no, it will be like we would have to start over. Well, I mean, we would have the RCB there, and we can continue and make an MCB, but all the testings would need to be done. So one clone cannot replace another. They're considered different, even though they're sister clones, in that sense. But clones also do behave very different than one another, which is the reason we're testing all of these clones. If they didn't, we wouldn't have to test that many, right? We could just pick one and save the trouble. So no, and quite often I think process development upstream and downstream would have to be redone and qualified as well. So it's definitely not that simple. You cannot just replace one with the other.
Thank you.
Yeah. Also, when we typically, so the reason that all of these steps are so critical in the whole like narrowing down the number of clones is that we prefer only making one MCB. So in some cases, I know Lily has generated more than one because the timeline was really of the essence. And then you can just, take your chances, generate a lot of MCBs and get that started. But it is a very expensive process to make the MCB and also to qualify it for release in GMP. So typically we don't advise doing that in more than one clone.
Thank you. All right. Here's a question referring to just scale of fermentation. So the question says that we only saw scales go up to 500 liters. For CHO cells, do you guys have a larger capacity than that for the single-use bioreactors?
I can answer that. I think Louise might be able to speak to the capability of Copenhagen site, but in Seattle, we do have a 6-pack, 6 times 2,000 liters. So that's our highest scale. We also have a site in Longmont, I believe, or Boulder, that can produce up to 20,000 liters.
Excellent. Thank you.
Sorry, I just had to step away to get some, battery charger to my computer. No, I don't have much to add. Sorry.
Okay, fair enough.
There is a question though on clone stability up to, I think, what would mean 120 consecutive generations, maybe. And typically, I mean, our stability studies don't run for that many generations. I think we would typically run them for now 9 weeks, corresponding to these 70 generations or so. And that would typically be our aim for the stability studies. So I'm not sure if we know about 120. Do we, Lily? Do you know anything about that? Like, have you done that long stability studies from cell line?
No, I have not done it for that many generations.
Okay.
Very good. Okay, well, thank you all for your questions and for your time today, for joining us. Please do reach out again to AGC if you have additional questions, and we'll certainly be following up with you after the fact as well. So thank you all for your attention. Thank you to our speakers. Have a great day.
Thank you.
Thank you.
Bye.